Question: have you ever wasted food? I think the answer for most people, including myself, is yes. Now, on to another question. Have you ever wasted $220 billion worth of food? Probably not. Although food waste by overzealous consumers like myself and perhaps you is certainly an issue, this can be controlled to an extent by improving food distribution and our individual purchasing choices. However, what cannot be so easily controlled are plant diseases, which cost the global food supply $220 billion worth of crop yield every year. Many of these plant diseases are bacterially caused. Pictured here are three such diseases. On the left, Pierce's disease affecting grapes by Xylella fastidiosa, that's the pathogen bacterium. On the top right, an apple tree infected with fire blight caused by Erwinia amylivora. And on the bottom right, an onion infected with center rot disease caused by various species of the Pantoia genus. This year, the UT Austin iGEM team is honing in on fighting bacterial plant diseases to mitigate agricultural losses. Now, after identifying bacterial pathogens as a major cause of crop loss, we decided to reach out to farmers to learn more about current solutions to the issue. We spoke extensively with Rising Sun Vineyard, a local vineyard that is impacted by Pierce's disease. The owners at Rising Sun told us that if even one plant were to become infected, they would have to purge the whole crop to prevent a total outbreak, which represents a major waste of time and resources. They and other stakeholders also told us that they regularly use pesticides to prevent insects from transmitting bacteria to the plants, but they expressed concern about the potential toxic effects of these chemicals on humans and the environment. Our discussions with Rising Sun and other stakeholders boiled down to one key finding: current solutions to bacterial plant pathogens are unsustainable.
And we wanted to use synthetic biology to solve this problem. Our goal was to develop a sustainable solution to bacterial plant diseases that is both modular and specific. To achieve this goal, we decided to engineer biocontrol Assa-Cins to kill pathogens using microcins. But let's break that down a bit. What are biocontrols? When researching sustainable solutions against plant pathogens, we came across bacterial biocontrols, bacteria that are naturally found in nature and can kill or inhibit a target pathogen. Despite their sustainability, however, biocontrols only account for about 1% of current agriculture control measures. This is in part because biocontrols are bacteria discovered to naturally inhibit a pathogen, so it can take a long time to find new ones. But what if we used synthetic biology to speed up the process by engineering biocontrols against specific pathogens? And that's where we decided to use microcins. Microcins are small antimicrobial peptides that certain types of bacteria naturally secrete to kill closely related species. Microcins are highly specific, mainly targeting close relatives of the bacteria that the microcin originated from. And because of this specificity, the chance of developing resistance is also thought to be lower for microcins than traditional chemical antibiotics. And finally, microcins are naturally found in bacteria and in the environment, and are therefore less likely to have off-target toxic effects than pesticides, thus making them more sustainable.
Now, to accomplish our overall goal of engineering biocontrols to fight plant pathogens using microcins, we established three objectives. Objective 1, use computational biology to screen for novel microcins against specific pathogens. Objective 2, create a modular system that allows for the biocontrol chassis to express and secrete a microcin of choice. I want to emphasize modularity here because we want to create a tool that is versatile and can be used to target a variety of different bacterial plant pathogens. And then finally, objective 3, assess the functioning of our modular system and effectiveness of our novel microcins through bioassays. But before we could begin identifying potential microcins in our pipeline, we had to first identify a model pathogen host system to test our proposed pipeline. This led us to onion center rot, a disease caused by various strains in genus Pantoia. We chose onions because rot can be observed in a matter of weeks rather than months, and they're easy to access at the store. And fun fact, onions are also the state vegetable of Texas, which allows us to have a little local impact as well. We also consulted with a multitude of plant pathologists and other onion rot experts to learn more about onion center rot and develop onion assays. To identify which strains of Pantoia to target with our microcins, we injected onions with 4 different strains of Pantoia thought to be pathogenic, and we identified Pantoia ananatus 97-1R as the most virulent. The figure you see here depicts an assay that we conducted to characterize the pathogenesis progression of this strain. As you can see on day 0, this is what a non-inoculated onion should look like. By 12 onward, however, the onions inoculated with Pantoia ananatus show severe signs of rot as compared to the water control shown on the bottom, which is showing little to no signs of center rot even after 12 to 21 days.
After establishing Pantoia ananatus as our main pathogen of interest, we moved to Objective 1, using computational biology to screen for novel microcins. Recently, a new open-source program called SINFUL was developed to identify potential microcins against specific bacteria by analyzing the genomes of their close relatives. We use SINFUL to search for potential microcins in the genomes of strains related to the bacterial pathogens that we were interested in. We compared genomic sequence data to a set of 10 known microcin sequences from E. coli, and specifically were able to predict 13 potential microcins for 3 genera known to cause plant disease: Pantoia, Erwinia, and Xanthomonas. Here's a figure of the results that we got on SINFUL when searching for potential microcin clusters against the Pantoia genus. The 6 main sequence clusters in this alignment tree gave us 6 potential microcins that we plan to insert into our microcin secretion systems.
Once we had identified potential microcins against Pantoia, we moved on to Objective 2, designing our modular microcin expression system. In the literature, we found an existing 2-plasmid microcin secretion system, with 1 plasmid containing the parts for exporting a microcin, shown on the left, and the other containing the parts to express the microcin, shown on the right. We wanted to redesign the existing system to be more modular, so we focused on engineering the microcin expression plasmid to have greater modularity in order to create a system in which microcins can be swapped in and out depending on the target pathogen. The original microcin expression plasmid and our redesign will be shown on the next slide. So this plasmid is a more detailed schematic of the existing microcin expression plasmid from the previous slide. You can see a signal peptide sequence on this plasmid. This is a short sequence required for the microcin to be exported by the cell. This second plasmid displays our redesign of the existing plasmid to increase modularity. We reorganized the plasmid so that the signal peptide is upstream of any microcin, which allows us to place any microcin or microcin plus immunity protein, as seen here, downstream for export. Our system can be applied to various pathogens, as potential microcins targeting any species can be found using SINFUL and incorporated into our modular plasmid. And now, over the course of our project, we made 13 microcin parts and tested 9 of these. We also made 7 inducible promoter parts, as well as parts for their associated transcriptional regulators. And we, of course, made a part for the signal peptide described just a moment ago, which was to be placed upstream of any microcin as seen on the previous slide. Characterization data for our 4 inducible promoters and their regulators can be seen on the results page of our wiki. As demonstrated in the following assays, we were able to confirm our microcin parts and signal peptide parts can work together as a cohesive unit to allow the expression of different microcins within our biocontrols.
And that allows us to reach the final step of our overall pipeline, which was to develop bioassays to test whether our modular novel microcin expression systems works in our biocontrols. We wanted to answer 3 main questions using these bioassays. First, can our biocontrols secrete microcins? Does our modular microcin expression plasmid work? And finally, do our novel microcins inhibit their targets? We went about answering these questions using a zone of inhibition assay. In a zone of inhibition assay, a predator strain is plated on top of a prey strain in soft agar. When a microcin is secreted by the predator strain, the prey strain should be killed and you will see a visible zone of inhibition. To determine if our biocontrol strain can secrete microcins in the first place, we tested a Pantoia biocontrol strain that is already used in agriculture and transformed it using the existing microcin secretion system in microcin V, which is known to target E. coli. You can see here that when the non-pathogenic Pantoja biocontrol is transformed with a microcin, there is a clear zone of inhibition where the E. coli prey has been killed. Once we successfully transformed our biocontrol strain, we moved on to our next objective, which was to validate our designed modular system using MCCV once again. Here, spot 1 is our modular system, spot 2 is the existing non-modular system, and spots 3 and 4 are our negative controls. From the zones, it appears that our modular redesign is about as effective as the original, and it has the engineered flexibility that will make it compatible with a variety of different microsins. We successfully confirmed that our modular system works in a zone of inhibition assay using MCCV. So our final goal was to test whether one of our 6 novel microsins that we screened using Sinful can inhibit their targets. We conducted a zone of inhibition assay using E. coli to secrete MCCO4, which is a potential Pantoja targeting microsin. Here, the top right has the control with MCCO4 and no secretion plasmid, and the bottom strain is a negative control. We observed a potential small zone of inhibition in the top left strain, which is E. coli that has been transformed with both MCCO4 and the secretion plasmid. However, this zone wasn't as strong as some of our previous zones, so we decided to characterize MCCO4 further. Specifically, we used a growth curve assay to monitor how our MCCO4 microsin affects the host Pantoja strain. The solid lines here are the average of all 8 replicates that we did. What's important here is the Pantoja strains containing the microsin but no immunity protein, which is shown in blue here, had the slowest growth, indicating inhibition of growth. But when we added the immunity protein with the microsin, which is the orange curve here, the Pantoja growth was not inhibited. This suggests the toxicity of MCCO4, our potential microsin, but we would need further analysis to validate these results.
In conclusion, over the course of our project, we established and completed 3 objectives. 1. We screened the genomes of multiple bacterial genera and identified 13 potential microsins and tested 9 of these for effectiveness against target pathogens. All of these microsins, as well as the various parts of our 2 plasmid secretion system, are in the iGEM parts registry. 2. We redesigned a microsin expression plasmid to improve modularity and make it easier for others to swap in and out their own microsins. Future synthetic biologists can adapt this system with various regulatory parts to secrete the microsins of their choice. And 3, we tested our microsin expression system using zone of inhibition and growth curves and found that our system allowed our biocontrols to successfully create and secrete microsins. All of our protocols and findings have allowed us to create an adaptable workflow that future iGEM teams and other researchers can build upon. But our work isn't done quite yet. We have certain next steps we'd like to take. First, we'd like to continue testing to confirm our novel microsins are actual microsins. Though the growth curve was promising for MCCO4, we need further validation to see whether our putative microsins can be secreted by our host organism. Second, we would like to perform an onion rot assay using our biocontrol Assa-Cin, engineered with a novel anti-Pantoja microsin such as MCCO4 to test whether it can prevent pathogenic strains of Pantoja from causing rot. And then third, we'd like to test expression of microsins using inducible promoters in our secretion system. We want to use inducible promoters to hopefully prevent toxicity to the host. Overall, our research has allowed us to create a system that gives farmers a modular, sustainable alternative to preventing plant disease. These results demonstrate the versatility and potential of microsins to combat important agricultural diseases and can also impact you as agricultural consumers to have access to more produce on store shelves. Thank you.